Our presenter will be Dr. Shishan Yuan, colleague from BI in the Research Center, and he will present some of the new ideas that may lead to a future vaccine for PRRS. Dr. Shishan is a PhD from the University of Minnesota. He worked there with Dr. Murtaugh, and please come on stage and teach us some of the new tricks in the PRRS world. Thank you.
Okay, first of all, thank you very much, Oliver and the organization committee, to give me this opportunity to give this talk, which probably would be my swan song for my academic life here, just because I joined the BI a year ago. Well, let me begin to give my thanks to the people who helped my academic career, and as you just heard, I did my PhD and the direction of Dr. Michael Malta and Kai Farberg in the University of Minnesota. And Yingfa has been quite helpful in sharing some of those reagents. Hans Nowick in Ghent University, and he is the one choosing us as a Asian coordinator for the EU PRRS project. And Eric Snyder and has been quite collaborative on some of those chimerical work we did on equine arteriviruses and PRRS virus. And Han Chun and Guang Zhi at Shanghai Veterinary Research Institute, and they have been quite helpful after I moved back to Shanghai in 2005. And without their help, I guess I couldn't get into those national key program. And last but not least, these are the other students, mostly of them are graduate students I coached during my 7 years academic career in Shanghai Veterinary Institute. They are the ones did the job, and after I left, and they jumped ship, and 8 of them are in the U.S. for further training. And by my count, 5 of them are actually working for BI now. So, well, what I'm going to talk about today is, well, the current situation, especially on the vaccine front, and then how much do we know about this PRRS virus, and based on that, and how we can move towards the next stage. And on the last part, I would tell you a little bit about we as a PRRS company, how we move on from here. And you heard a lot excellent description what happened during 2006 from Dr. Yang just earlier. So what I'm trying to emphasize is the high fever disease is an overly used to generalize term in the field now. Everything is high fever disease, but well, it's being controversial. What is a high fever disease? Or you blame PRRS is the only reason. Well, in looking back, when we trying to find the main causative agent, what the several main lab, what we come up with, the top 3 agents are actually PRRS, CSF, and PCV2. And remember, in the West, PCV2 causes PCVAD. That's exactly the same time on the other side of the world. In looking back, and the PCV2 actually is one of the major part of the so-called high fever disease back then. And then we also have all those traditional good, bad CSF merged in those infections. So to me, PCV2 was overlooked at the beginning. And then second part is what we call high fever disease is a bucket of co-infecting agent. And then, well, it's been controversial about the pathogenicity of this virus. To me, it's another hard PRRS virus, just like MA184, the different strain. But, well, as you might have heard, and the Vietnam strain has been shipped to Plum Island and Cresa in Spain, and they couldn't reproduce the clinical observation we observed here in Asia. And fortunately, and as you may heard at USDA, Kelly and Cal reproduced the study using our Chinese PRRS, and it seems like the infection of the Chinese PRRS indeed can induce higher level of secondary infection. And on the vaccine front, and as Dr. Yang just pointed out, we probably got too many, too few of the good vaccines. And you heard from Mike, cured vaccine, believe it or not, there is still a market here, and even in Southeast countries, and including some of those are actually top one. And the BI vaccine was the first registered modified law vaccine in the Chinese market, and 2 years after, and the MLV based on classical strain of the Chinese PRRS also licensed, and during this hot topic of high fever disease, and we got 3 more MLVs that's based on the so-called high pathogenic PRRS. And the red one, including this cured vaccine, are actually on the Chinese government tender business. And where do we go from here? And that it's quite murky, and sometimes some farms use a vaccine and still you get this disease, so-called high fever disease. And part of the problem is maybe we have too many choice on those vaccines with variable quality. And then in the end, and where is the next generation novel vaccine you can come up with? Overall, what you look at efficacy, cured vaccine is not insufficient as a protective immunity. Well, you can rule out some of those, and then safety, as Dr. Yang indicated, some of those vaccines is actually artificial inoculation. You actually get disease from those vaccines. And by all said and done, and DWA feature by which you can differentiate your herds with vaccination or natural infection. That would be a desirable feature for the next generation of vaccine. For PRRS, if we want to do this, how much do we know? Well, there is a Chinese saying, and you would win the battle 100% of the time if you know your enemy and yourself. Unfortunately, in my mind, we don't know much about the PRRS. And PRRS started as a mystery disease, and still some puzzling part that make us scratch our head. And as a matter of fact, and for the past 2 decades, and people believe, and this inside of this virus is an icosahedral nuclear capsid, which is until 2009, somebody using a novel method by the name of cryo-electronic microscopy. It's actually showed we were wrong for 2 decades. Actually, it's a helical nuclear capsid. And then we're wrong on this front, and then people have been putting too much effort on those big proteins. And the major protein, just because in the virus particle, the amount, the portion are bigger and are higher. But there are tens of different minor proteins. Are these minor protein plays just a minor role, or we have been misled by this simple, the size of the protein? And then the upper panel is a genomic structure, and we haven't seen the last yet. Well, I'll tell you a little bit. Actually, the structural gene here is overcrowded, and there is actually hidden gene in this genome. So PRRS virus has numerous tricks we don't know yet. So we need to solve those tricks before we even talking about next generation. Well, one of the relatively novel method we use is reverse genetics. The reverse genetics, the way you do it, at least for virus research, you take out the virus genome from the virus particle and convert it into a DNA copy by which you can use a scissor to create the kind of mutation you desire. And then this could be infectious. You transfect those into the cell and you get a recombinant virus. And part of the effort in my previous life, well, academic life, has been trying to decipher what are the critical elements that control the life cycle of this virus. And as you heard from Dr. Yuan, and they have been busy working with trying to which part of this virus actually is a virulence determinant. Being in industry now, my goal is we can create the next generation of vaccine based on this. Well, based on what we learned about this virus, we can use rationally design for the next generation of vaccine. What I'll present to you today is part of the work have been doing in my previous lab at the Shanghai Veterinary Research Institute, and one is what is the deal about the glycosylation? Can you actually use it to play with the virus to reach the goal? Another part is if you believe GP5 is the main protective antigen, can you increase the expression level of this virus? And the last part is, well, we are trying to search which is the target for the virus. So we just talked about the major protein that's long believed is a major target for the past 2 decades. Well, could still be, but we were missing the real target. Well, the first part is, as I just mentioned, glycosylation of a protein has been believed is a trick by the virus to shield your host immunity. So-called this overglycosylation that has been reported in influenza virus, in HIV, and the overglycosylation just block the reaction of the neutralizing antibody against the virus. And on the other part, an glycosylated protein could be an integral part of your antigen. So you get a double-edged sword. So how do you play with this? To begin with, what we started is we want to check out the biological significance of this individual glycosylation site, which can be predicted by using a computer program. And actually, Mike's lab has been using biochemical methods to identify those different glycosylation sites. And this glycosylation site actually has been discussed for a long time, and people actually using a different glycosylation pattern to categorize different viruses. So actually, we don't know much of the biological significance. What we did is we take out every one of these signals so that you don't get glycosylation for the virus particle, or a combination of two, including we take all the glycosylation site. And then what you can see here is if you take one or more of those glycosylation sites, the virus can still be viable, including this one. Just has no glycosylation site, which means this glycosylation actually is not essential for virus viability. So that's none of the glycosylation site, and actually including the one has been reported here at position 44. This was reported 10 years ago, claimed that this position is critical for virus replication. It's turned out, I'll touch it a little more, and they actually changed a hidden gene instead of the glycosylation site. Well, to conclude this part is, we know this glycosylation site is not necessary. If we believe the overshielding of the glycosylation site, overshielding against the host immunity is true, then we can take that off in the future vaccine candidate. Well, the genetic code for all this protein, you could not change the left part of the protein. This is roughly the ectodomain part. And if you change it, the virus won't grow. But you can change the right-hand part. And unfortunately, this right-hand part are the internal cytoplasmic domain. I mean, this is it's not exposed, it's indoor, it's in contrary to the ectodomain. So that's not what we want. We were scratching head at that time as to why we can change the right hand. We could not change the left hand. And this is one almost field study until, let me just first show you the experimental data. And here, if you change everything, or the left part, you don't get virus. The rest, you can play around as you want. And then in one of this Asian PRRSpective meeting, and I sat down with Mike, and Mike was just find the ORF5A that's using another novel method by the name of mass spectrometry. It's turned out in the virus particle, actually there is other structural protein that's in the virus particle. But previously, if you use a traditional biochemical method, you wouldn't identify this. Well, then that's answered our question why we cannot change the left-hand part. We could change the hidden gene. Well, we use a genetic method. We knock out actually the hidden gene, which called the ORF5A now, in 2 North American strain and in top 1, which is European strain. And all of those, as you can see, if you knock out this hidden gene, you wouldn't have a viable virus that comes out with it. So this is says the hidden ORF5A actually is an essential part of the virus infection cycle, which somehow in contradictory with what reported by Eric Snyder for EAV system, where they found, well, it's not essential, but you need it. Is that the virus was crippled to a 2 log low of virus titer. But well, that's what we did, and in the available system, we found this is actually essential for the virus particle formation. Well, what this do, we know it's essential, and we have been trying to figure out what role this small protein may play in the process of infection. And we did some small experiment before I drop all this. And what we found is if you supply this ORF5A in trans, and actually you can make this virus infectious for another round. which means this small protein could play a role in the process of virus binding the virus into the cell and have to get out. And this would be where this small protein play the role. Well, altogether, and we confirmed what Mike found is an essential part of this virus. It's another part of the trick the virus played we don't know yet. Even on those newly written publication or textbook, and that's the genomic map, what you think is actually wrong. It's not the complete picture yet. Well, the last part, the technical part, what I want to talk about is, well, where is the main target for control of this, or in other words, what the host immunity plays a game against this virus. And we want to address this question is, well, we can use different species, as you know. Arterivirus, Arteriviruses has 4 different viruses that can infect pigs, horse, and the monkey and mice. These are the only 4 different arteriviruses. And we can actually create chimeric virus. Previously, we done study, and we can create chimeric virus between top 1 and the top 2 pairs, and then we make one step further. Let's create some virus between the horse and pig virus, by which we can identify some of the key determinant for control of the virus infection or play a role during this process. And as this is another part, and the main this is a Chinese name, is actually the viral attachment protein. This protein is actually responsible for recognize the cell, the target cell, to get into the cell, to engage with the receptor. It's sort of lock and key. Well, you have to, this target has to be there first. But as I said, for the past 2 decades, we all believe this GP5, the big O protein, was the one to target the target cell. And then last year, biochemical data from Nebraska showed actually the minor protein are the ones interact with the CD163, which is believed is a core receptor for the virus get into the cell. Well, and what we did is, like I said, this is a PRRS virus infectious cDNA clone, this is the equine virus, and we put the equine virus small protein, the minor protein, into the background of the PRRS virus. By that, we create a chimeric, and we were quite fortunate, and we actually got the infectious virus out of it. And as you can see, this is a PRRS virus with a minor protein of the equine virus. And as you can see, it's viable, and it's becoming, this recombinant virus express the GP2, the minor protein from the equine virus. And then this virus recombinant, as we all know, PRRS virus can only infect primary cell like alveolar macrophage, and there is only one monkey kidney cell line MA104 or derivative. And PRRS virus can only infect this one. But for equine virus, they can infect all 3. So the chimeric virus that with a minor protein from the equine virus be able to infect the rest of the cell. So that says the minor small protein we put into the PRRS virus backbone actually determine the cellular tropism, which means, well, the virus data showed here, which the minor protein played the central role, probably in finding those target cells, well, we talked about, as you heard from Mike, and the neutralizing antibody. We don't know how big a role that will be played, but where the neutralizing antibody would target, is target the virus entering the cell. This is the stage the neutralizing antibody played a role at. And so by finding this minor protein actually is an attachment protein, the neutralizing antibody actually should direct to this smaller one. Instead of GP5, you heard the previous significant money have been put into the investment, DNA vaccine, subunit vaccine, vectored vaccine, all use GP5. So the GP5 actually could be the wrong target you are using. If you are developing the subunit vaccine, my suggestion is drop it, stop it. And this is the target. The minor protein actually is a major target. Well, the last part, when I jumped the ship and being in the industry, and as I said, we are the first company, and we have been devoted a huge resource on how to improve to the next vaccine. What I'm going to tell you is, and the kind of investment Boehringer Ingelheim have put into the Chinese, as a matter of fact, Asian market. And actually, the animal health related investment comes into two parts. And one, we finally we have an inauguration ceremony in Shanghai. Some of you are the distinguished guests in Shanghai. And we have a functional lab now. And on the ceremony, this is our board member, Mr. Feng Baumach, and my boss and the global head for R&D. And Dr. Yuan is here, and Professor Chengping Lu was there. And as a matter of fact, Dr. Yang and a group of the swine virologists were invited at the ceremony. And then another significant part is we will have a factory. We will have a manufacturing site in Taizhou. And this, we just had a ceremony for the joint venture with the local Taizhou CMC government. And this manufacturing site is expected to produce a product with roughly 2 years down the road. And a little bit more about the R&D site. This R&D is roughly 2,000 square meters, and we will reach to the head count of 60 scientists in Shanghai within 2 years. And then we will have a process development department in that group too. Right now, we are standing at 28. Actually, our target actually is 38. So here is an advertisement message. We are recruiting. Some of those positions actually is quite hard. Well, as you heard from Ham, I guess sometime, and you Chinese school don't have a professional vet. Yes, in a way, and the graduate students like to do molecular biology. And well, we think that we cannot do necropsy pathology, and that's my message to you, my colleagues. If you know somebody, or just drop me a line. We need that help. And we will have a veterinary trial center in Taizhou too, and that will be up and running next year. We will do swine and poultry, and it's quite a big investment. A little bit more about our global R&D, and before 2009, our main campus is in the US, in the Midwest. And then after 2009, we purchased the footage for the pet vaccine development. Of course, close to the headquarters in Ingelheim, we have a Hanoi site that actually will be inaugurated the day after tomorrow, based on what I know. So you heard about the Shanghai site, and we altogether have a 12 R&D site, and it's a global organization. And we in Shanghai, the Shanghai site actually is well supported by the global. We have 450 scientists altogether, and then we rely on heavy collaboration for the upstream work and some of those clinical development. And as I said, we get a little team now, and we have fun sometime after work. And those are some of those pictures on our site, and this is a building, and we are actually 5 kilos away from the famous pond. And this is inauguration, and this is one of the lab for molecular biology. And this is the office area. And in Shanghai, believe it or not, you can find some European style site. And this is our team in the lab. And 100% of those equipment are imported from the Western world. So on facility-wise, we actually are invited by our global partners. Okay, with that, I'll stop. And okay, yeah.
Do we have any questions for Dr. Yuan? No questions? I have a quick question for you, Shishan. You talked about chimeric vaccines for PRRS, and I presume that would be a live vaccine. Will you have some concerns about safety with a live chimeric vaccine?
Well, first off, the chimeric vaccine has been, well, at least close to the market now, as you may heard. And CSF vaccine based on the backbone of BVDV actually is close to registration in Europe now. This is actually the same scheme, same strategy. And this virus, regarding to safety, we don't know yet. Well, what we got is actually a virus, a chimeric virus, we have not put into the animals. What you can expect from this is, well, could be replicative, could be infectious in the animal, but what we hope is it's just a single round infection. So, and to answer your question, we don't know what to expect, but our best bet is, well, this vaccine could be in between the killed virus killed vaccine and the live vaccine. You have the safety of killed vaccine, and you have the efficacy from MRV. So everything remains to be answered when we put into the animals.